Hospital-associated infections are a major challenge for clinicians around the world. These pathogens and a subset of specific opportunistic pathogens have learned to cause infection outbreaks within the hospital-built environment. These infections can impact the most vulnerable in our population, including the very young, like newborn babies, the very old, cancer patients, or anybody who comes into a hospital with a compromised immune system. Studying and tracking these infections and modelling these infections in a lab is very difficult to do. There's relatively few, if any, models that can accurately recapitulate the actual transmission and spread of bacteria within the hospital-built environment. With this new Galeria melanella hospital-associated intensive care unit model that we have developed, we have shown that we can 3D print replica intensive care units and use these intensive care units and Galeria melanella as vectors to spread infection to track infection spread around the hospital-built environment. This gives us one of the first in vivo models to be able to track bacterial dissemination through a built environment. This gives us untold opportunities to study the genetic determinants that are associated with bacterial dissemination through the built environment, but we've also shown that this model can be used to study and develop novel antimicrobial surface coatings, which may help to tackle the antimicrobial or the hospital-associated infections within hospitals around the world. The model can be used for high throughput screening of novel surface coating formulations and then can be used to determine if they are effective or ineffective in preventing or attenuating the spread of infection through the hospital-built environment. We believe this model, which is incredibly portable, versatile, and can be established in almost any lab around the world, represents a unique opportunity to gain insights into bacterial dissemination through a built environment, but also to fast-track the development of novel antimicrobial surface coatings. Sterilize the ICU 3D model using 70% ethanol for a minimum of 90 seconds and allow it to dry in the sterile environment prior to use. If you are testing a surface coating, saturate a cotton swab in 1 ml of the coating solution. Use the swab to coat the outer walls of each room. Re-moisten the swab and use it to coat the inner and outer wall of the central room. Repeat this until virtually all of the 1 ml solution is used up, whilst resaturating the swab every 3 to 4 rooms. Layer the Petri dish with foil sheets topped with filter paper to elevate the bottom of the dish to ensure that the top of the 3D model is touching the Petri dish lid. Randomly select 6 instar larvae and sort them into Petri dishes lined with filter paper. Allow 10 caterpillars per dish and store them at +4 degrees Celsius until use. Use 70% ethanol to sterilize the larval surface prior to the experiment. Place a single sterile larvae into each patient room of the ICU model. Place a 1 cm squared agar media patch with your pathogen of interest grown on it into the middle room of the model. Incubate the setup at 37 degrees Celsius for 24 hours. After 24 hours incubation, remove all larvae from the model and euthanize them by placing them at -20 degrees Celsius. Move the ICU model to a new sterile Petri dish with a sterile tweezers. Use a sterile PBS-soaked cotton swab to swab the walls of each room to collect the bacteria. Break off the swab tip into a 0.5 ml of sterile PBS and vortex. Serially dilute the collected samples and plate them on nutrient agar plates. Incubate the plates for 18 hours. Enumerate the colony forming units after the incubation period.